editing of CCR5 in autologous CD4 T cell of person infected with HIV. Definition: AHB is an lentivirus that causes the acquired immunodeficiency syndromes, a condition in humans in which progressive failure of the immune system allows life-threatening, opportunistic infection, and cancer to thrive.
HIV, or human immunodeficiency virus, is a virus that targets cells in the immune system. Over time, the immune system begins to fail, which is called immunodeficiency, and this increases the risk of infections and tumors that a healthy immune system would usually be able to fend off. These complications are referred to as AIDS, or acquired immunodeficiency syndrome. Now, there are two distinct types of HIV, HIV-1 and HIV-2. HIV-1 is more commonly associated with AIDS in the US and worldwide. HIV-2 is more rare and typically restricted to areas in Western Africa and Southern Asia. HIV-2 is so uncommon that HIV almost always refers to HIV-1. Alright, HIV targets CD4 plus cells, meaning cells that have this specific molecule called CD4 on their membrane. Macrophages, T helper cells, and dendritic cells are all involved in the immune response and all have CD4 molecules. Therefore, they can be targeted by HIV. The CD4 molecule helps these cells attach to and communicate with other immune cells, which is particularly important when the cells are launching attacks against foreign pathogens. So this little molecule is pretty important for our immune system, but it's also extremely important for HIV. HIV targets and attaches to the CD4 molecule via a protein called GP120, found on its envelope. HIV then again uses GP120 to attach to another receptor, called a co-receptor. HIV needs to bind to both the CD4 molecule and the co-receptor to get inside the cell. The most common co-receptors that HIV uses are the CXCR4 co-receptor, which is found mainly on T cells, or the CCR5 co-receptor, which is found on T cells, macrophages, monocytes.
Application is nursing. The goal of anti-AAV gene therapy is to produce an immune system with cell resistant to viral replication, which are both functional and fit infection by other organisms.
We are now going to discuss the immune system and the basic functions of the elements that comprise the immune system. The blood is the major organ in the body that comprises the immune system and its ability to ward off infection. The body has many types of blood cells. Chief among these are the white blood cells that are themselves divided into several subcategories. The white blood cells are those which are directly responsible for fighting off infection, and they comprise a number of subsets. White blood cells are responsible for immunity, and they are responsible as well for the body's defense mechanisms that fight infections that are a variety of different types. These include bacteria, fungi, protozoa, and viruses. Phagocytes are a major type of cell in the body responsible for immunity as well. These are responsible for the engulfment and consummation of foreign particles. The phagocytes literally can eat foreign bodies that include viruses. Lymphocytes are a major component of the immune response. These are responsible for specific immune responses that can be divided into several types.
Pros and cons. The long-term success of the therapy is based on the expectation that infected lymphocytes will be destroyed, allowing those modified with resisting CCR5 to proliferate and becomes the predominant CD4 population.
So now we're in the lymph node. We're looking at how HIV, brought to the lymph node by either one of our well-intentioned dendritic cells or maybe by a macrophage, manages to infect our T helper cells, aka our CD4 lymphocytes. Well, it all sort of comes down to receptors. On our T helper cell membranes, we have 2 to 3 receptors that help HIV gain access to these cells. We have the CD4 receptor, which is sort of the primary receptor that HIV needs, and we have either one or both of CCR5 and CXCR4. CXCR4 being much less prevalent. CCR5 is really the preferred co-receptor that most strains of HIV need to bind. But these are both called co-receptors in this context here. So what's important about these particular receptors is that HIV happens to have a protein, this thing here called GP120, on its envelope, the outer part of the virus here, that interacts really well with these receptors. You can almost think of it as a lock and key setup. These receptors here are some of the locks that need to be opened to gain access to our T helper cell, and unfortunately for us, HIV has the key, this GP120 protein on its surface.
Used split in Ecuador. On November 25, in Quito, he made known to the world the creation of the first genetic modified human. Beijing used the Cas9 technique. Between the base of the world science communicate and contradiction unquestionable technical and biological science.
So it's my great pleasure to introduce the final two speakers, and the first one is Paula Kynan. And you heard from the end of this morning the sort of development of the incredibly rapid development of CRISPR-Cas9 technologies, right from the discovery through to its ubiquity or its usefulness in various systems, through then to thinking about the types of applications, and including gene drive in mosquitoes and disease control in the environment. You're going to hear from Paula Kynan today about the work that she's done as a pioneer in trying to apply this type of technology to human diseases, in particular to HIV/AIDS and the treatment of prevention, actually, of stopping the HIV virus in its tracks. And it's interesting in looking back at how rapid this technology has become mainstream. And you heard it a little bit this morning, but there was a casualty, and the casualty was the TALEN technologies.
Conclusion. CCR5 modified autologous CD4 T-cell infusion are failed within the limit of the study.
So we know that in an untreated HIV infection, the levels of the virus rise up. They rise up to extremely high levels throughout our bloodstream and throughout our body. And that's because HIV is getting into our immune cells, right? They hijack our T cells and they use them to replicate and to make lots and lots of copies of themselves. But we also know that we see this massive drop-off in our T cells, our CD4 cells in particular. So the question is, well, how is this happening? I mean, yeah, we see that these cells get infected, but why are so many of them dying? Do they all get infected? Do they die when they get infected? Well, this question is one that scientists have been working on for decades, probably about 30 years so far, and it turns out that in an HIV infection, only about 5 to 10% of the immune cells that end up dying off are actually HIV infected. The remaining 90 to 95% that die off are not even infected. That's ridiculous, right?